Hello everyone, today I will be discussing about genetic manipulation in plant. Genetic manipulation in plants is like giving plants a little makeover at the genetic level. It involves changing specific genes to make plants better in some way. This technology has opened up a whole new world of possibilities in agriculture and plant breeding. Importance of plant biotechnology or genetic manipulation in plant is crucial for several reasons. Food security, if we see the food security, by improving crop yields and making plants more resistant to pest and disease, we can ensure that there is enough food to feed a growing global population. In case of environmental benefits, genetic modification can help reduce the need for harmful pesticides and promote more sustainable farming practices, which is good for the environment. In nutrition, we can enhance the nutritional content of plants, which is essential for combating malnutrition and providing healthier food options. For economic growth, improved crops can lead to higher agriculture productivity, boosting the incomes of farmers and supporting economic growth. If we see the medicinal plant, genetic manipulation can also be used to produce valuable medicine from plants, making healthcare more accessible. DNA and genes. Overview of DNA and genes in plant cells. Inside plant cell, there is a remarkable molecule called DNA, which stands for deoxyribonucleic acid. Think of DNA as the instruction manual for building and operating a plant. It is like the blueprint that tells the plant how to grow, develop and function. Now, DNA itself is quite long and consists of millions of tiny units called nucleotides, which are strung together unlike a chain. These nucleotides are represented by the letters A, T, C and G. It is the specific order of this letter that makes up the genetic code. How genes determine plant traits? Genes are like individual chapters in the plant's instruction manual. Each gene has a specific job or set of instructions. These instructions are coded in the sequence of nucleotides within the gene. For example, let us say there is a gene responsible for producing a protein that gives a plant blue flowers. If this gene is turned on actively producing the blue color protein, the plant will have blue flowers. If the gene is turned off or not functioning correctly, the plant may have a different color of flowers like red or yellow color. Plant genetic variation. Natural genetic variation in plants like all living organisms come in a variety of shapes, size and colors. This natural diversity among plant is due to something called genetic variation. Genetic variation is like the spike of life for plants. It is what makes each plant unique. Introduction to genetic diversity. Genetic diversity is like the variety of flowers in an ice cream shop. It refers to the range of different genes and traits found within a population of plants. This diversity is incredibly important for several reasons. For example, imagine a field of wheat with different varieties. Some may be tall and resistant to drought, while others are shorter but more resistant to disease. if a dry year come, the taller drought resistant wheat will do better. If a disease outbreak occurs, the disease resistant wheat will thrive. This diversity help ensure a more reliable food supply. Genetic modification versus traditional breeding. Traditional breeding and genetic modification are two different methods for improving plant traits, and they each have their pros and cons. If we see the traditional breeding, so you can see how it works. In traditional breeding, plants with desired traits are selected as parents and their offspring inherit a mix of genes from both parents. Over many generations, breeders select the offspring with the best trait and continue breeding them. It is a time consuming. Traditional breeding can take many years or even decades to develop new plant varieties because it relies on natural process of gene mixing and selection. Limited precision. It is challenging to control which specific genes are passed on to the next generation. So, there is limited precision in trait improvement. For example, developing a new apple variety with a specific taste and color through traditional breeding would involve planting and selecting apple trees for many years, hoping that desired traits eventually emerge in the offspring. Genetic modification. If we see the genetic modification, you can see how it works. Genetic modification involves directly changing a specific gene in a plant to achieve the desired trait. Scientists can add, delete or modify genes with precision. We can get the faster result. GM can lead to faster result compared to traditional breeding. Scientists can introduce a desired trait into a plant in a relatively short time. Precision. GM allows for precise control over the genetic changes, which can be especially useful for traits that are difficult to achieve through traditional breeding. If we see the example, creating genetically modified corn that produce a toxin to protect itself from pest. This can provide better pest resistance compared to traditional breeding methods. Advantages of genetic modification. Genetic modification offers several advantages in plant improvement. For example, in precision, GM allows scientists to precisely target and modify specific genes responsible for desired traits. This precision can be challenging to achieve through traditional breeding. Speed. Genetic modification can lead to quicker result compared to traditional breeding methods, which can be time consuming. Access to new traits. GM can introduce trait from unrelated species, opening up new possibilities for improving crops. For example, adding genes from a frost resistant fish to a plant can make it more tolerant to cold temperatures. Reduced environmental impact. GM can lead to plants that are more resistant to pests and diseases, reducing the need for chemical pesticide and promoting more environmentally friendly farming practices. Nutritional enhancement. In the case of nutritional advancements, genetic modification can be used to enhance the nutritional content of crops, addressing issues like vitamin deficiency in certain regions. crop resilience. GM can help create crops that are more resilient to adverse environmental conditions such as drought, salinity or poor soil quality. Increased crop yields by improving trait related to yield such as disease resistant or drought tolerance, GM can contribute to higher crop yields and increased crop production. Now, techniques for genetic manipulation. What are the techniques? So, overview of common genetic manipulation techniques, CRISPR-Cas, GMOs, etc. So, there are several common genetic manipulation techniques used in plant biotechnology to modify plant DNA and improve desired traits. Here, we will provide a brief explanation of each technique. We will take one by one. First is the CRISPR-Cas technology. What is CRISPR-Cas9? Is a revolutionary gene editing tool that acts like molecular Caesar. CRISPR is a guide molecule that directs Cas9, a protein, to a specific location in the plant's DNA. If we see its working, how its work. So, once at the target location, Cas9 cuts the DNA. When the cells repair the cut, it can introduce desired changes or edits to the gene. So, what are the advantages of this technology? So, the advantage of this technology is highly precise, efficient and versatile for making specific changes in the plant gene. Next is the genetically modified organism, GMOs. What are GMOs? GMOs are plants that have had their DNA altered using various genetically engineering techniques. This alteration can involve editing, deleting or modifying genes. If we see its function, how its work, so scientists introduce specific genes into a plant's DNA to impart desired traits such as pest resistance, herbicide tolerant or improved nutritional content. And its advantages are rapid development of traits including resistant to pest and diseases and the ability to adapt to challenging environments. Then RNA interference, RNAi. What is RNA? RNA interference is a mechanism that regulates gene expression by preventing the translation of specific genes into protein. If you see the function of it, how it works, so the small RNA molecules are introduced into the plant which bind to messenger RNA or mRNA and prevent the production of corresponding protein. So, its advantage is it can be used to suppress harmful genes or control gene expression in plants, helping them resist pest or produce desired compounds. Next is transgenesis. What is transgenesis? So, transgenesis involves the introduction of genes from one species to another. It is a common method used in the creation of genetically modified plants. It is a specific genes often from unrelated species and inserted into the plant's DNA. These foreign genes become a part of the plant's genetic makeup, and its advantages are it allow the introduction of entirely new traits or characteristics that may not naturally occurs in the plant species. Then selective breeding. What is selective breeding? Selective breeding is a traditional method for genetic manipulation that involves choosing plants with desired traits as parents for the next generation. if we see its working, so how its work? Over generation, plants with the desired traits are repeatedly crossed to concentrate those traits in the offspring. And its advantages are, while not a modern biotechnology technique, selective breeding has been used for centuries to develop new plant varieties with specific traits. Next one is the marker assisted breeding. What is marker assisted breeding? Marker assisted breeding combines traditional breeding with genetic information. It helps identify and select plant with desired traits more efficiently. Okay? Its working is, genetic markers like specific DNA sequences are used to identify plants that carry the desired genes without the need for extensive phenotypic screening. So, its advantage is, speeds up the breeding process by targeting specific genes of interest. So, benefits of genetic manipulation in plants are, the genetic manipulation in plants offer numerous benefits contributing to advancement in agriculture and addressing various challenges. Let us explore some of the key advantages, including improved crop yields, pest resistant, enhanced nutritional content and environmental benefits. Improved crop yield also. So, how it works? So, the genetic manipulation can enhance the ability of plants to produce more and higher quality fruits, grains or vegetables. Its advantages are, the increased crop yields help meet the growing global demand of food. This is especially critical in regions where food scarcity is a concern. Farmers can produce more food with fewer resources, leading to greater agriculture efficiency. And pest resistance also. In pest resistant, genetic modification can make plants resistant to pest and disease by introducing genes that produce natural toxins or enhance the plant's defense mechanism. So, what are the benefits? It reduces damage from pests and diseases, means less need for chemical pesticide. This not only saves farmers money, but also reduces the environmental impact and potential harm to non-target species. It also contributes to more sustainable farming practices. Then enhanced nutritional content. Enhanced nutritional content, genetic manipulation can increase the level of essential nutrients, vitamin and minerals in crops. So, what are the benefits of it? In this process, plants with improved nutritional content can help combat malnutrition and provide better access to vital nutrition, especially in regions where dietary deficiencies are common. For example, golden rice is genetically modified to produce higher level of vitamin A, addressing vitamin A deficiency in some populations. Then environmental benefits. In environmental benefits, genetic manipulation can lead to more environmentally friendly farming practices by reducing the need for chemical fertilizer, pesticide and herbicide. And the benefits of this environmental process is, decrease use of chemicals, reduces soil and water contamination, benefits non-target organism and promotes sustainable agriculture. Additionally, genetically modified crops designed for drought tolerance or salt resistant or thrive in challenging environments, helping to conserve water resources and protect fragile ecosystem. Reduce food waste. So, genetic manipulation can extend the shelf life of fruit and vegetables or improve their resistance to brushing or spoilage. So, its benefits are it reduces food waste, means less food is discarded due to spoilage, damage or deterioration during storage or transportation. This is economically advantageous and help address global food security challenges. Then specialty crops. Genetically manipulation can create specialty crops with unique traits such as ornamental plants with vibrant color or bioengineering plants that produce pharmaceutical compounds. So, what are the benefits of it? If we see the benefits of these plants, so specialty crops have been various application beyond food production, including the pharmaceutical and horticultural industries. They can provide novel products and genetic economic opportunities. Genetic editing versus genetic modification. Genetic editing and traditional genetic modification are two distinct approaches to altering the genetic makeup of organism, including plants. They differ in how they make changes to an organism's DNA and the precision with which those changes are achieved. Now, distinguish between gene editing and traditional genetic modification. So, the gene editing, often using technologies like CRISPR-Cas9, involves making precise changes to specific genes within an organism DNA. In precision, it offers a high degree of precision, allowing scientists to target and edit specific genes with great accuracy. So, what are the outcome of it? If we see the outcome of it, the changes made through gene editing are typically subtle and involves modifying existing genes rather than introducing entirely new genes. For example, repairing a defective gene, enhancing an existing trait or disabling a harmful gene within a plant's genome. So, what are the traditional genetic modification methods? So, the traditional genetic modification involves introducing foreign genes, that is transgenes, into an organism DNA to confer specific traits or characteristics. It is generally a less precise than gene editing and it involves editing or altering entire genes, sometimes from unrelated species. The introduced gene may encode entirely new proteins or function that are not naturally present in the organism. So, this is the outcome of it. If we see the examples, introducing a gene from a bacterium into a plant to make it resistant to insect pests or adding a gene from a different plant species to enhance its nutritional content. So, the pros and cons of each approach in gene editing. So, what are the pros? Gene editing is highly precise, allowing for the targeted modification of specific genes without introducing unnecessary genetic material. It can be faster process than traditional genetic modification, resulting in quicker development of desired trait. It reduces regulatory burden. Some regulatory agencies treat gene edited organism differently from genetically modified organism, potentially streamlining the approval process. So, what are the cons? Off target effect, although highly precise, gene editing can sometimes cause unintended changes in the genome known as off target effects, which needs to be carefully monitored and minimized. What are the ethical concerns in it? The ability to make precise edit to the genome raises ethical questions, particularly when it comes to human gene editing. So, the complexity are the gene editing techniques can be technically complex and require expertise in molecular biology. So, what are the pros in traditional gene modification? It is a versatile. Traditional genetic modification allows for introduction of a wide range of genes and traits into the organism. Traditional genetically modification has a track record of success in developing genetically modified crops with valuable traits like pest resistance. The outcome of traditional genetic modification are more often more predictable and well understood. What are the cons? Cons are it is less precision, it is generally less precise than gene editing, potentially resulting in unintended effects or disrupting native genes. The regulatory approval process for GMOs can be lengthy and costly, involving extensive safety assessment and public scrutiny. So, what are the public perception? Genetically modified organisms have faced public skepticism and regulatory hurdles due to concern about safety and environmental impact. Disease resistant and stress tolerance. Disease resistant and stress tolerance are critical traits in plants that determine their ability to survive and thrive in various environments. Genetically modification plays a significant role in enhancing these traits, contributing to improved crop health and yield stability. So, genetic modification for disease resistant, scientists can introduce genes into plants that confirm resistance to specific diseases. These genes often come from other plants, microorganisms or natural sources. Enhancement of plant defense. Genetic modification can also boost a plant's natural defense mechanism, making it more effective at recognizing and combating invading pathogens. In some cases, genetic modification is used to reduce the population of disease carrying insects or other vectors, indirectly reducing disease transmission. Enhancing the stress tolerance in plants. So, genetic modification can enable plants to conserve water more effectively, withstand extended periods of drought and recover more rapidly when water becomes available again. Salinity tolerance. By introducing genes that help plants exclude or detoxify salt ions, they can become more resistant to saline soil, which are common in arid region. Genetic modification can improve a plant's ability to tolerate extreme temperature such as cold or heat, allowing them to grow in regions with challenging climates. Now, real world application and success stories. We all know about the Bt cotton, Bacillus thuringiensis. So, the Bt cotton is one of the most well known example of genetically modified crops for best resistance. It produces a protein toxic to certain insect pests, reducing the need for chemical insecticides. Golden rice So, the golden rice is genetically modified to produce higher level of provitamin A, that is beta carotene, addressing vitamin A deficiency in many parts of the world. This enhances the nutritional content of rice, making it a powerful tool against malnutrition. Another example is disease resistant papaya. In Hawaii, genetically modified papaya was developed to resist the papaya ring spot virus, that is PRSV, papaya ring spot virus. This saved the Hawaiian papaya industry from devastation and allowed farmers to continue growing this important crop. Then drought tolerant maize. So, genetic modification has been used to develop maize varieties that are more resilient to drought condition. These varieties help ensure food security in regions prone to water scarcity. Another examples are salt tolerant rice. So, the researchers have engineered rice varieties that can thrive in salty soil. This has the potential to expand rice cultivation to regions affected by soil salinity. Next example is heat tolerant wheat. Genetic modification has been applied to develop heat tolerant wheat varieties, which are essential as global temperatures rise due to climate change. So, with this I conclude today's lecture. We have learned about genetic manipulation in plant, how they are introduced in the plants, their application in various sectors, and some of the real world examples of product of genetic manipulation. I hope you are enjoying these talks and feel free to communicate if you have any queries with you. Thank you.